Are you an anthropology enthusiast or a UGC NET aspirant aiming for success? You've landed in the right place. Welcome to ANTHRONET Academy. At ANTHRONET Academy, we specialize in simplifying anthropology concepts, tackling tricky MCQs, and guiding you through comprehensive preparation strategies for UGC NET and beyond. With engaging content, insightful lessons, and a community of dedicated learners, we're here to make your journey not just easier but exciting. Subscribe now and take the first step toward mastering anthropology and achieving your academic goals. Unit 2. Topic 2.2. Theories of Evolution. 1. Contents. Cope's rule. Dallow's rule. Allen's rule. Bergman's rule. Gloger's rule. Haldane's rule. Synthetic theory of evolution. Neutral theory of molecular evolution. Cladogenesis. Anagenesis. 2. Cope's rule. Founder Edward Drinker Cope. Definition: over evolutionary time, the body size of animals within a lineage tends to increase. Examples: evolutionary trends in horses and dinosaurs show increasing body sizes over time. Exceptions: some species evolve smaller sizes, e.g., island dwarfism. 3. Dallow's rule. Founder Louis Dallow. Definition: evolution is irreversible. Once a complex trait is lost, it cannot be regained in its original form. Examples: loss of limbs in snakes or eyesight in cave-dwelling species. Exceptions: some studies suggest partial reversals may occur under specific conditions. 4. Allen's rule. Founder John Allen. Definition: animals in colder climates have shorter appendages, ears, tails, limbs, to conserve heat, while those in warmer climates have longer appendages for heat dissipation. Examples: Arctic foxes, short ears and tails. Desert foxes, long ears for better heat regulation. 5. Bergman's rule. Founder Carl Bergman. Definition: within a species or closely related species, body size tends to be larger in colder climates and smaller in warmer climates to optimize heat retention or dissipation. Examples: polar bears, large body size to conserve heat. Tropical bears, smaller body sizes for better cooling. 6. Gloger's rule. Founder Wilhelm Lambert Gloger. Definition: animals in humid regions tend to have darker pigmentation compared to those in arid regions, likely as a form of protection against UV radiation or camouflage. Examples: darker plumage in tropical birds. Lighter fur in desert mammals. 7. Haldane's rule. Founder John Burden Sanderson Haldane. Definition: in hybrids, if one sex is absent, rare, or sterile, it is usually the heterogametic sex, e.g., XY in mammals or ZW in birds. Examples: male sterility in mule hybrids, horse and donkey. The synthetic theory of evolution, also known as the modern synthesis, integrates Charles Darwin's theory of natural selection with Gregor Mendel's principles of heredity and advances in genetics, paleontology, systematics, and biogeography. It provides a comprehensive explanation of evolution as a process driven by multiple factors. Key features of synthetic theory. 1. Genetic variation. Variation arises through mutations, recombination during sexual reproduction, and gene flow between populations. These genetic changes provide the raw material for evolution. 2. Natural selection. Differential survival and reproduction of individuals based on heritable traits. Traits that provide a survival or reproductive advantage become more common in the population. 3. Gene flow. Exchange of genes between populations through migration. Increases genetic diversity and prevents divergence between populations. 4. Genetic drift. Random changes in allele frequencies in small populations due to chance events may lead to the loss or fixation of alleles over time. 5. Reproductive isolation. Barriers to gene flow, geographical, behavioral, temporal, lead to speciation. Populations evolve independently, resulting in new species. 6. Speciation. Gradual accumulation of genetic differences leads to the emergence of new species. Can occur through mechanisms like allopatric, sympatric, or peripatric speciation. 7. Role of fossils. Fossil records provide evidence of gradual changes over time and transitional forms linking different groups of organisms. Contributors to the synthetic theory. 1. Darwin, natural selection as the driving force of evolution. 2. Mendel, principles of heredity and genetic inheritance. 3. Fisher, Haldane, and Wright, mathematical models of gene frequencies and natural selection. 4. Dobzhansky, population genetics and genetic variation in natural populations. 5. Amer, species concepts and reproductive isolation. 6. Stebbins, evolution of plants and genetic mechanisms. 7. Simpson, role of fossils in understanding macroevolution. Significance. The synthetic theory of evolution provides a unifying framework, connecting microevolution, small-scale changes within populations, and macroevolution, large-scale evolutionary changes and speciation. It remains a foundational concept in evolutionary biology, though modern research has expanded it to include epigenetics, developmental biology, and molecular genetics. The neutral theory of molecular evolution, proposed by Mitsuo Kimura in 1968, emphasizes that most evolutionary changes at the molecular level are not driven by natural selection but by random genetic drift of neutral mutations. These mutations have little to no effect on an organism's fitness. Key concepts of the neutral theory. 1. Neutral mutations. Mutations that neither improve nor harm an organism's fitness. These mutations accumulate over time due to random genetic drift. 2. Genetic drift. Random changes in allele frequencies in a population, especially significant in small populations, causes neutral mutations to become fixed or lost over generations. 3. Molecular clock hypothesis. The rate of molecular evolution is approximately constant over time for a given gene or protein. Neutral mutations provide a clock to estimate evolutionary divergence times between species. 4. Selective constraints. Most functional genes and proteins are under selective constraints, limiting the types of mutations that are neutral. Regions with low selective constraints, like non-coding DNA or synonymous sites in coding sequences, exhibit higher rates of neutral evolution. 5. Types of mutations in the neutral theory. Synonymous mutations do not alter amino acid sequences and are mostly neutral. Non-synonymous mutations alter amino acid sequences but are often neutral or deleterious. Evidence supporting neutral theory. 1. Molecular variability. High genetic variation observed at the molecular level, inconsistent with strong natural selection alone. 2. Relative constancy of molecular evolution rates. Molecular clock studies show relatively constant rates of mutation accumulation across lineages. 3. Polymorphism. Patterns of genetic variation within populations often match predictions of neutral evolution. Criticism and modern perspectives. 1. Role of selection. While the neutral theory argues most changes are neutral, natural selection still plays a significant role, especially in adaptive evolution. 2. Nearly neutral theory. Tomoko Oda, 1970s, proposed that many mutations are nearly neutral, having small effects on fitness that drift or selection may influence depending on population size. 3. Modern genomic data. Advances in genomics show that while much of the genome evolves neutrally, regions under strong selection, e.g., coding regions and regulatory sequences, are exceptions. Significance. The neutral theory shifted the focus of evolutionary biology to include stochastic processes like genetic drift and emphasized the importance of neutrality in molecular evolution. It complements the selectionist view and remains a cornerstone for understanding molecular evolution and population genetics. Cladogenesis refers to the evolutionary process by which a lineage splits into two or more distinct species, leading to an increase in biodiversity. It is a key mechanism of speciation and contrasts with anagenesis, which involves evolutionary changes within a single lineage without branching. Key features of cladogenesis. 1. Branching evolution. A single ancestral species diverges into two or more descendant species. It results in the formation of a clade, a group of organisms that includes an ancestor and all its descendants. 2. Reproductive isolation. For cladogenesis to occur, populations must become reproductively isolated, preventing gene flow. Isolation can be geographical, allopatric speciation; ecological or behavioral, sympatric speciation. 3. Genetic divergence. Isolated populations accumulate genetic differences due to natural selection, genetic drift, and mutations. Over time, these differences result in the formation of distinct species. 4. Increased biodiversity. Unlike anagenesis, which replaces one species with another, cladogenesis creates new species, contributing to the diversity of life. Examples of cladogenesis. 1. Darwin's finches. An ancestral finch species colonized the Galapagos Islands and diversified into multiple species adapted to different ecological niches. 2. Mammalian radiation. After the extinction of dinosaurs, mammals underwent cladogenesis, evolving into diverse groups like primates, cetaceans, and rodents. 3. Cichlid fishes. In African rift lakes, cichlid fishes have diversified into hundreds of species, each adapted to specific feeding habits and habitats. Mechanisms leading to cladogenesis. 1. Allopatric speciation. Populations are geographically isolated, e.g., by mountains or rivers, leading to divergence. 2. Sympatric speciation. Populations diverge within the same geographic area due to ecological or behavioral isolation, e.g., different mating preferences. 3. Peripatric speciation. A small population becomes isolated at the edge of the larger population's range, leading to rapid divergence. 4. Parapatric speciation. Populations adjacent to one another diverge due to gradients in environmental conditions. Table of difference between four types of speciation. Importance of cladogenesis. Biodiversity. Cladogenesis explains how new species arise, contributing to the variety of life forms on Earth. Phylogenetics. Understanding cladogenesis helps reconstruct evolutionary relationships and build phylogenetic trees. Adaptive radiation. Cladogenesis is often associated with adaptive radiation, where species rapidly diversify to exploit different ecological niches. By creating branching lineages, cladogenesis is a primary driver of the evolutionary tree of life. Anagenesis refers to a pattern of evolutionary change within a single lineage where one species gradually transforms into another over time, without branching into distinct species. This process results in the replacement of the ancestral species by the descendant species, with no splitting of the evolutionary line. Key characteristics of anagenesis. 1. Linear evolution. Evolution occurs in a straight line within a single lineage. 2. No speciation. There is no branching. Instead, one species evolves into another. 3. Gradual change involves the accumulation of small, incremental changes over time. 4. Fossil record, often represented as a sequence of transitional forms in the fossil record. Anagenesis is contrasted with cladogenesis, where an ancestral species splits into two or more distinct species. While anagenesis represents evolutionary change within a lineage, cladogenesis emphasizes the diversification of lineages. Example. An example of anagenesis is the evolutionary transition from Australopithecus afarensis to Homo habilis within the hominin lineage, where gradual changes in morphology and behavior led to the emergence of a new species without splitting. Upcoming topic: 2.3 Trends in primate radiation. Thank you for watching. We appreciate your time and support. Subscribe to Anthronet Academy for more engaging content on anthropology. Please like, share, and comment on this video to help us grow. Stay connected. We're here to support your UGC NET exam journey to success.